Hello students, this is the Distinction Learning Platform, and on this platform you find illustrations, explanation to difficult topics, as well as examination revisions as well. So a lot of stuff that you're going to find. Before we begin our lessons, make sure that you subscribe so that you can be updated before upcoming videos. To make the story short, let us begin. Okay, so this is the third part of cell injury, and we're talking about atrophy. Okay, remember that in the previous lessons we talked about hypertrophy, which is basically the enlargement of the cell, the cell size. All right, so now this is the second part now, which is we're talking about atrophy under cell adaptation. Just to remind you, remember that cell adaptation is what? It's a reversible change of the cell if it is exposed to the change in the environment. All right, so now what about now? So one of the cell adaptation is atrophy. Remember that what is atrophy? Atrophy is the decrease in the size of the cell resulting in the decrease in the size of the organ. Okay, what is atrophy? Atrophy basically we are saying is a decrease in the size of a cell resulting in the decrease in the size of an organ. Okay, so we'll talk about the morphology of how a cell can look like if you look at grossly or if you look at under a microscope. So look at that will be under morphology. Okay, so now what else? Also, make sure that you understand to say, why do you think cells undergoes atrophy? Why do you think cells undergoes atrophy? It's because, okay, it is because a cell has decrease in the functional demand as well as decrease in the workload. That's a major reasons why cells undergoes atrophy. Or the bigger reason is because a cell has undergone it is experiencing decrease in workload as well as it is experiencing decrease in functional demand. Remember that if we talk about the workload, from the previous videos we described to say workload is the amount of activities or amount of work that is given to the cell. And what about functional demand is the amount of activities that is required for a cell to perform. Okay, now in this case we're having decrease in functional demand, we're having decrease in workload. Okay, now what could cause a cell to experience decrease in functional demand and also to experience decrease in workload? What could cause that? So there are basically two causes of that. There is what we call physiological causes and also pathological causes. So if we talk about causes, there are two. Physiological causes, basically these are the normal causes of the cell, mean that it's not because of a disease. So it's not because of a disease, it's because of normal circumstances that are happening in the body. All right, so that are happening in the body resulting in a normal response. But under pathological causes, it's because of an underlying condition such as a disease and a cell is undergoing atrophy. Okay, so we'll start with physiological causes of atrophy. So what is the first physiological cause of atrophy? Remember that there is what we call embryogenesis. Embryogenesis. Okay. Under embryogenesis, I can give a good example of where atrophy occurs under embryogenesis. Remember that embryogenesis, that's where embryo develops. Remember that there are a lot of structures that are going to be developing inside the embryos. So a good example is the development of the thyroid gland. Remember that the thyroid gland begins the development of the thyroid gland. It will start from the mouth and it has to go down to the neck. So now there is a structure that will connect there is a structure that will connect the mouth and also the neck. That structure has to regress, and that structure, that's where the thyroid gland has to pass from. Okay, but after the thyroid gland passes from, it has to regress around the 10th week. What do we call that? That structure in the during embryogenesis of the thyroid gland, we call it thyroglosal duct. So under embryogenesis, we can just talk about the regression of the thyroglosal duct. So regression of thyroglosal duct. Okay, so that's under the physiological cause of thyroid. So mean that for that thyroglosal duct to regress, it has to undergo atrophy. Okay, because if it was left persistent, it can cause developmental defect, like causing fistulas, etc. So that's why it's a normal physiological process. It starts to regress. Okay, what about the other cause? So this is basically in fetus. Then what about in adults? In adult, what is the physiological cause of atrophy? So remember that in the adult there is what we call aging. Okay, so that's a physiological process, meaning that that's a normal process. So during aging, remember that there will be atrophy of the what? Of the brain. So in adult, we are just basically talking about aging. So that's a physiological process. So there's going to be atrophy of the what? Atrophy of the brain, atrophy of the gonads, okay, and as well as atrophy of the heart. So this is basically the normal physiological cause of what? Atrophy. In infants or in embryo, there will be during embryogenesis there will be regression of thyroglosal duct. In adult there will be aging. Then there will be what? Atrophy of the brain, atrophy of the gonad, and also atrophy of the heart. So those are the physiological causes of what? Atrophy. Now, what else? Let's go to the pathological causes of atrophy. What are the pathological causes of atrophy? There are a lot of pathological causes of atrophy. Okay, I'll start with the first one. What is that? So, disuse. We can just say disuse of limbs. Okay, so what are the situations where someone can't use their limbs? It's because maybe they had a fracture. So a fracture, that could be a pathological cause why someone cannot move. If someone cannot move, they are not using their limbs, the cells, they will respond by being smaller in size because they are not moving. So what do we call that disuse of limbs? We call it immobility. The first cause is being immobile for a longer period of time, then atrophy will be there. Then what else apart from the disuse of limbs or immobility? Then what else? The loss of innervation. Okay, loss of innervation or denervation. Okay, so that is the second cause of atrophy. So basically, all cells has to be innervated by nerves so that they can be, what can I say, communicating with one another. They can be like being activated. Nerves has to be there. But what if there is a condition that can cut off the nerves? Okay, like poliomyelitis. So that condition can cause nerves not to connect to a cell. If nerves are not connecting to the cell will respond by what? Being smaller in size, so that at least they can what? They can decrease further the functional demand and also decrease the workload. So in that way they can try to survive. Otherwise, minus doing that, minus being smaller in size, what can result? It can result in cell injury or cell death. Okay, so that is loss of innervation. What else? Also, we can also say ischemia. Okay, ischemia can also cause atrophy. What is ischemia? Ischemia basically is what? Is the decrease in the blood flow to end organ. Okay, so decrease of flow of blood to different organs, that is what we call ischemia. So as long as if you look at there's decreased perfusion to different tissues or organs, then that is what we call ischemia. So now if there's decreased blood flow to the organs or tissues, now there will be decreased transport of nutrients, transport of oxygen, transport of a lot of important substance to a cell. Now if a cell recognize that there is decrease to that, then how is it going to respond? It's going to become smaller inside, so that those little nutrients that are coming to the cell, they are going to be enough or sufficient for that cell because it is smaller inside. So that's how ischemia can cause atrophy of different tissues. Okay, what else? There are a lot of causes. Okay, what else? Okay, let me make the story short. Let me just put one other good example or good cause of atrophy or pathological cause of atrophy, which is this starvation. The last important one, starvation. So if someone is starving, basically they are lacking nutrients, important nutrients to the body. So this is a general cause. These are all local causes up to ischemia. Starvation is a generalized cause, meaning that it can affect almost all body tissues. Okay, if someone is starving for a longer period of time, the body cells, they are experiencing low nutrients in the body. Basically, so those nutrients, since a cell need nutrients in order to survive, how do they respond? They need to respond by what? Reducing the size, so that those little nutrients that are going to those cells, they can also be sufficient. Why? Because the cell is what? Smaller. Okay, that is under starvation. These are all pathological causes of atrophy. All right. So now let's go to another session. So since we know the causes, we know what atrophy is and why there is atrophy. The reason why there is atrophy, remember that there is the functional, there is decreased functional demand, and also there is the decreased workload. Okay. So now we'll go to this part talking about the mechanism of atrophy. So we need to know under cellular level what happens, how does a cell reduce its size? Like in hypertrophy, we looked at the mechanism on how a cell becomes large in size. Remember that atrophy is just the reduction of a cell size, resulting in the decrease in the size of the organ. That's what we said. That's what I said. Okay. So now mechanism. So mechanism of atrophy is similar to hypertrophy, but basically it's just in the opposite way. Atrophy, reduction in size; hypertrophy, increase in the size. Okay. If you look at the mechanism of atrophy, basically what are we looking at? This one has two important combination of mechanism. Remember that atrophy, we are going to say the first one is what? Reduction in what? Protein synthesis. What else? And the other one is increase in protein degradation. So these are two important mechanisms in which a cell can reduce its size. Is either it doesn't produce protein, because proteins are the major component of a cell. If you increase protein synthesis, mean that you are going to increase the size of the cell. If you decrease the protein synthesis, then you are going to reduce the size of the cell. Now, either way, if you reduce the protein synthesis, yes, basically it's going to reduce the size of the cell. If you increase protein degradation, mean that the rate at which the protein are being broken down, it is increased. So since you are breaking down proteins, you are not going to have enough protein in a cell. Then what's the result? The result is what? A reduction in the size of the cell. Now, this is just the overview of how the mechanism of how a cell can undergo atrophy. Now, let us look at the cellular mechanism leading to decrease in protein synthesis and leading to increase in protein degradation. Okay, let us start with this one. Remember that this receptor, the first receptor, this one, we call it a growth receptor. So the first one, this one, is called the growth receptor, and that's where growth factors binds to. Remember the types of growth examples of growth factors we said: insulin-like growth factors, fibroblast growth factors, transforming growth factor beta. Those are the examples of what? Growth factor. They always bind to the what? Growth factor receptor, so that they can induce signal transduction pathway. So that's the first one. Now, in this situation, if you don't need protein synthesis, you're going to decrease the binding of growth factors to the what? Growth factor, or to the growth factor receptor. So you're going to decrease this growth factor to bind onto what? Growth factor receptor. Why? Because of these causes that we talked about. That's the reason why there's a reduction in growth factor, like binding to the growth factor receptor. Okay, what else? There is also this one. Remember this one from the hypertrophy lesson. We said this is a mechanical sensor. So this one is a mechanical sensor. This is what we talked about. Mechanical sensor. Okay, remember that mechanical sensor is, how is it activated? It is activated because of what? Remember that it is because of stretch, mechanical stretch. Why? Because there is increase in because there is increase in what? Weight load. But here there is decrease in what? Weight load. So then what do you expect on this part? There is what? Decrease in what? Weight load. There is decrease in activation of what? Mechanical sensor. Basically, there is a reduction in the what? Mechanical stretch. So if there is reduction of mechanical stretch, there will be reduction in the activation of what? Mechanical sensor. What else? Remember here we said these are what? Hormone. So this is a hormone receptor. Basically, we called it under hypertrophy, we said hypertrophic receptor. Here we're going to call it atrophic receptor. But let's just call it hormonal receptor, because that receptor responds to what? Different hormones, alpha adrenergic hormones, like agonists, like epinephrine, bradykinin. They respond to endothelin 1, angiotensin 2. So there are a lot of substances that are going to bind to that agonistic receptor. Okay, again, you're not going to find those things here. There will be reduction of those things. Okay, so everything, regardless of any stimuli, is going to be reduced. So if there is a reduction of all these three things, what do you think will happen? So there will be reduction in the what? Signal transduction pathway. So there will be reduction in the what? Signal transduction pathway. If there is a reduce in the signal transduction pathway, what do you think will happen? What will happen is, now there will be reduction in the activation of what? Transcription factor from what? DNA. Okay, so if this is the DNA, so the transcription factor, activation of transcription factor from there will be what? Reduced. So reduced activation of transcription factors. Reduce activation of transcription factors. If transcription factors are not activated, what do you think will happen? There will be no translation. There will be reduced what? Protein synthesis. So what do you think if there is a decrease in the transcription factor activation? There will be decrease in what? Translation, and decrease in what? Protein what? Synthesis. So as you can see now, that's how decrease in protein synthesis comes about, because of all these things that are coming about. So this is molecular mechanism. This is what happens. Okay, and why do you think this happens? Because of these causes. And there is also another pathway. Another pathway is what? Leading to increase in what? Protein degradation. And this is a very, very important part. Make sure that you concentrate. Okay, so now how does it lead to increase in protein degradation? So degradation means what? Breaking down protein, because you don't need proteins under atrophy. Okay, so what do you think will happen? Yeah, remember that because of these causes, physiological or pathological causes, it activates certain substances in the what? in the cytosol or in the cytoplasm of a cell. Okay, if the cell wants to undergo what? Atrophy. So what am I simply saying? If someone is exposed to either physiological causes or pathological causes, it's going to activate something here inside the cell. So what do we call that? It's going to activate certain enzymes. So we've got E1 enzymes, E2 enzymes, and the E3 enzymes. So remember, what's the first thing? Because of those causes that we talked about, physiological, pathological, what do? They are going to activate what? Certain enzymes in the what? Cytoplasm of a cell. Like what? Enzyme E1, enzyme E2, and enzyme E3. So specific name for enzyme E1, we call it ubiquitin activating enzyme. That's the first E1 enzyme. E2, that is ubiquitin conjugating enzyme. The last one is what? Ubiquitin ligase, which is the E3. Those are examples of what? Enzymes that are going to be what? Activated. Okay, so these three enzymes, what do? Okay, there's a very, very important protein that you need to understand, which is what we call ubiquitin. Ubiquitin. So this protein ubiquitin, what does it do? Ubiquitin. So ubiquitin is a protein that consists about what? 76 amino acid. And what's the major importance of that protein in a cytoplasm? So that protein, that ubiquitin, targets what? Cells, or tags cells for what? Degradation. If a protein, like let's say, if this is a protein that has to be what? Degraded in the what? In this vesicle, then ubiquitin has to be what? To be there. So ubiquitin has to go to what? If this red substance ubiquitin has to go to the what? The protein that needs to what? To be degraded. Now the question is, how does ubiquitin go or tag itself or find itself in the what? Protein that needs to be degraded? It's because of what? The help of these three enzymes. So these three enzymes, they help to transfer ubiquitin to what? The protein that has to be what? Degraded. All right. So what am I simply saying? So E1, E2, and E3 enzymes, what do? They are going to what? Transfer what? Ubiquitin, okay, to the protein. So if those ubiquitin proteins have been transferred to the what? Protein that has to be degraded, they mean that protein has what? Has been tagged for what? Or tagged for degradation. So once that happens, then that protein that has to be degraded, it will be what? Transferred into this vesicle. Okay, this vesicle is called what? They call it protosome. Okay, protosome. So that vesicle that I've drawn from there, that is what we call a protosome. So a tagged protein is going inside the protosome. That's where degradation what will occur, because inside the protosome there are some degrading substances. Like there are a lot of enzymes that are going to degrade that protein, a lot of substances. So substances that can be found inside the protosome, those are the substances that will help to break down the protein that has been tagged because of the help of what? E1, E2, E3 what? Enzymes. What are the names of E1? E1 is what? Ubiquitin activating enzyme. E2 is what? Ubiquitin what? Conjugating enzyme. E3 is what? Ubiquitin ligase enzyme. So those are the three enzymes that are going to help to tag the protein for the degradation. Okay. Now once that protein now is inside there, it's going to be degraded inside there. Then it's going to be what? Degraded into smaller particles, or smaller substances. What do we call those smaller substances? Remember that now there are two. There are two substances that are going to be produced there. The first one is, we know that is what? Amino acids. Okay? The second one is what? Peptides. So two important substances that are going to be what? Produced after degradation of what? Tagged proteins, because of the help of three enzymes. Okay, now remember that the important substance that are going to be is what? Amino acid and peptide. And peptide they will be further converted into amino acids. Okay, now you can ask to say what we call this whole pathway starting from this enzyme up to the what? The degradation of what? The enzyme. What we call that pathway? They call this. So it's a bigger word. Okay, we call it ubiquitin proteasome pathway. Okay, ubiquitin proteasome pathway, the pathway that I'm talking about. So remember that now this whole pathway, remember that we are now degrading proteins. This is now, it will lead to what? Increase in protein what? Degradation. So this whole thing is leading to what? This whole thing is leading to increase protein degradation. That whole thing is resulting in what? Increase in protein degradation. Now the other question that you can ask is, why producing amino acid, or where do amino acid go, and why do we produce amino acid? If this cell wants to undergo atrophy, but why producing amino acid? So because amino acid, if someone is undergoing atrophy, if the cell is undergoing atrophy, basically it's because of what? A lot of causes. So let's just talk about one cause, which is starvation. So let's see now, where do you think amino acid go? So they go to the liver. If someone is starving, remember that they are experiencing what we call hypoglycemia. Basically, they are having no glucose levels in the blood. So how do the liver respond? So okay, there are other organs that can also produce glucose, but the major organ is the liver. So amino acid will rush to the liver. Then in the liver, it will help in the production of what? Glucose, by the process called gluconeogenesis. Yeah, so that's what that will happen in the liver. So that's the reason why this amino acid will go to the what? To the liver, to help in further production of amino acid, in further production of what? Glucose. Why producing glucose? Because someone is starving, they are having no energy. But if you're having more glucose, then at least you're going to have more energy production. Okay. So now let's talk about the morphology. How can you see a cell if it has undergone atrophy? How can you see it? You can see it in two ways. Whether using your naked eyes, you're going to say that is gross. Gross basically means you're using your naked eyes. Then gross eyes, you can't see a cell gross. So if you are looking at a cell using your naked eye, you can't see a cell. You can only see what? You can only see an organ. So based on what I'm simply saying is, on gross morphology, you're going to see increase in the size of an organ. In the decrease size of the what? An organ. On the microscope, mean that you're using now an instrument microscope. So on microscope morphology, what can you see? Decrease in the size of a cell. Okay, so that's what you can see. So that's where we end from on morphology, what you can see using your naked eyes and using microscope instrument. Now in the next video, we'll talk about hyperplasia in details as well, causes, physiological causes as well, and pathological, and lastly a mechanism of hyperplasia. Okay, thank you for your time, and make sure that you subscribe so that you can be updated with all upcoming videos.